So the question that I had for the panel is thinking about the importance of timing. And I don't know much about biological clocks, but I know there's some thought that telomeres may be a type of biological clock that helps the cell know how old it is. And so my question is the following. If a cell would normally shorten telomeres over time, does it make a difference when the telomere shortening occurs at the wrong time? So in other words, one of these early life stressors that would cause telomere shortening. So A, is it likely the cell knows that it's too early? And B, if it does know it's too early, what is the clock that it might use to know that?
I could try that unless anyone wants to go first.
They all look like they would welcome that, Alyssa.
I think it's a really fascinating question, and I don't think we have a great answer. But from what I understand, there is somewhat of a threshold effect of telomere shortening. So when we look at average telomere length and we find that it's short across cells, that means there are more cells in the blood that are senescent, that have telomere length that has gotten to a critically short length that has changed the functioning of that cell. And so when telomeres dysfunction, they uncap or they get too short, they send these DNA damage signals and signals to the mitochondria. That whole pathway has been mapped out, that whole cell signaling pathway. The mitochondria then dysfunctions and looks like a very, very old machine, that of an elderly person, where it is now leaking reactive oxygen species. So now you have a cell with short telomeres and full of oxidative stress. And not only that, we also know that the cell, senescent immune cells, particularly CD8 cells, start secreting pro-inflammatory cytokines. And we know from Judy Campisi's work in fibroblasts that when the cells start secreting cytokines to its local environment, it creates a very fertile ground for cancer.
Yeah.
So the short answer is, if there are enough senescent cells, you're changing a young person's biochemical milieu to an older person through systemic inflammation and poor immune function. We have a paper that's about to be in press. It's by Sheldon Cohen, and it showed that in young, healthy people, telomere shortness predicts infection to the cold. And this was a controlled study where people were put in a hotel quarantine and infected with a cold virus. And the people who had short telomeres in their CD8 cells had an 8-fold risk of getting full clinical colds. So there is definitely a clinical implication of having shorter telomeres in young, healthy people.
Thank you, Alyssa. Thea, you wanted to comment?
I agree with what she said. I would just add one thing to it. I think the time is important, but also the place. Because if you're a lone cell that has a shortened telomere, because there's been a break or many of the things that David spoke about, you're a lone voice in the wilderness, and you can yell, and it's a short response. But if there's many of you, and you're all sending out the DDR signals, the damage or the senescent or the stress signals, then there's strength in numbers, and you cross a threshold, and now it cascades and goes beyond.
So if I could just do a brief follow-up question then to Thea. I'm also wondering about, in your stochasticity, if a cell has an inappropriately shortening telomere, whether it recognizes that and tries to upregulate telomerase in a way to send it back in the other direction, and whether or not that could be one of the drivers, if it happens at the wrong time in a normal cell, for reactivating telomerase.
We haven't studied that, so I don't know the answer to that question.
Maybe I'll go. Lou Gross at University of Tennessee. So this is a follow-up to a comment that Alyssa just made about infectious disease, only she made it in the reverse. The question is there any evidence for chronic infections, Hep C, HIV, and or in women, for example, chronic yeast infections, of impacts on telomere length? Anything like that?
Yeah, I can try to answer that. There is, some emerging evidence in Epstein-Barr virus and immunocompromised patients, and that there's upon EPV infection, and also mono patients, which is a nice model for that, where you see changes at the telomere, essentially, happening and DNA damage responses going on with components leaving the telomere and going to those DNA damage responses. So there is some emerging evidence of that.
Not a lot, though, it sounds like.
What?
Is this an area in which there really does need to be more sort of investigation, collaboration on units that are dealing with infectious disease in particular?
Absolutely, yeah. I'm going to go to Stony Brook after this with a collaborator who works on EPV, and it's a really interesting area.
Joyce Suji, exponent and committee member. I wanted to thank the speakers for some really stimulating talks. And I think what really intrigued me was all the different interconnections the speakers were talking about. And I think Dr. Sandler in particular emphasized how difficult it is sometimes to understand how are these things relating, and is telomere shortening an end result, or is it a causative factor in the disease outcomes? And it's probably both, from what it all sounds like. And that being the case, when you have situations like looking at epidemiological correlations between many different pesticides and telomere length or even disease outcomes, how do you make any sense out of what you just found? I mean, you found 4, I guess, insecticides and 2 herbicides in which they were correlated with shorter telomeres. But is there anything about those chemicals? Can you classify the type of stress they're producing, or anything else that would bring some sense into what type of cause and effect relationship is there?
Excuse me, I think it's, really early in looking at this, and we are trying to, make sure that, at the simplest epidemiologic level, that what we're not seeing is one pesticide, but people use 2 or 3 or 4 of those together. I think various oxidative damage mechanisms are relevant to all of those, the pesticides that we found, as well as inflammation, but I don't really have a clue at this point. We're the first ones to report this, so.
Sounds like fertile ground for more research.
Hi, my name is Dennis Harris. I had questions about lifestyle and changes in telomere length. And it's actually 2 questions. The first is, can you explain how they exclude pre-existing genetic differences in the length of telomeres in different lifestyles? So whether a person meditates or takes multivitamins daily, or whether they're smokers or alcoholic or obese. And can you tell me whether or not changes in lifestyle result in differences or changes in telomere shortening rate? So if I were to quit smoking, does it change the rate of telomere shortening? Or if I were to take up meditation but still smoke, does it change?
I'll comment after Dale. Dale might want to comment.
I say those are terrific questions, and we are just, we've just started scratching the surface. And, it's the same thing with a new toy. So epidemiologists just discovered that they can have this relatively high throughput assay and can start, looking at things in this arena. And no, those kinds of studies haven't been done. But that's what's needed. We really, just looking at this cross-sectionally at one point in time, it's intriguing, but we need to look at, what happens to people who change things. I can imagine we'll find a clinical trial sooner or later.
Yeah, that's a great answer. I think the nice thing about the genetic variation of telomere length is that there is convergence across these GWAS studies, and we've been working on that now currently in Haynes. So there are, a handful, around 10 genes that do reliably predict telomere length, each contributing a tiny bit, though. It's clearly, polygenetic, but it does give us really nice opportunity to look at gene environment or gene behavior interactions, and that has not been done yet. It's been done with depression. A group has looked at actually a gene for MAO synthesis and depression, and found that genetic vulnerability for depression plus exposure leads to shorter telomeres. But anyway, in terms of your question about how do people control for genetic variation, they don't, not yet. And I think the action will be in the interaction and looking at them as a moderator. And then your question about what if I'm a smoker and an exerciser or meditator, and those are really important questions. And I think that we will be very much in the dark if we just look for main effects and covary out everything else in our model and look at bivariate relationships, because really behavior comes in these clusters, and bad things go together. But then there are these buffering factors that can interact, especially exercise. There's a paper by Eli Putterman that just replicates our finding with perceived stress, showing that women high in perceived stress, older women, have shorter telomeres if they're sedentary. But if they're exercisers, if they're doing daily vigorous exercise, they look like low stress women. So exercise can buffer the stress-induced telomere shortening, it looks like. And that paper, that model of lifespan telomere length that I flashed up, the whole point of that paper is that there are these multi-system resilience factors. Like Dale mentioned, the idea of resilience, where we won't find main effects of stress or exposure, at least very weakly and only in large samples, but we may find we find large effects of severe stress, but if we really want to understand stress in daily life, we need to also look at the positive side, the buffers, which is social support, stress resilience, and lifestyle factors. And the net effect of exposures plus the buffers will give us the best understanding of current risk and telomere length, I think.
This is Shortening. I have a follow-up question for the panelists related to this. Is that recently there's a finding showing that there are gender differences between the happy gene, which is so happen to be also related to MAO. So the question is, it only for women. So they found a happy gene for women, which is, this MAO synthesis. And so the question is, why is it not happening in men? I mean, are you guys having a slightly more complex psychology that we have to figure out? So. Since nobody wants to touch it, I actually want to bring this up. I think I want to look at this. There are reproductive stages in women that are quite discrete, and so perhaps whereas men, in theory, can father till 105. So I think that not that. I think male doesn't have andropause, but I think there are discrete reproductive stages. So maybe that is one of the reasons why we are not really looking at a two-dimensional interaction, gene and environment interaction. We're actually looking at a three-dimensional interaction, that is gene, environment, and life stages interactions. So many of the things that are set out early in life or at a particular stage of life will need a manifestations down the road, like a pre-programmed life stage play out. So maybe the panelists can address that in the context of telomere or any other, sort of chromatin complex structures. So I'm just making a comment. So we have two more people right over there.
Valerie Adams, U.S. Army Public Health Command. Insert the disclaimer here that the questions and comments are my own. And I think my question might follow up on Dr. Ho's, because what I want to know is, a cell spends most of its time in interphase. It's not actively dividing. And in that relaxed chromatin state, what is going on with the telomeres? David mentioned that he's maybe going to be collaborating with somebody looking at viral interactions. And it seems to me that that would be the prime time for virus to be interacting at the telomeres is during that relaxed chromatin phase. And so what is the state of the science for that?
I think it's more to do with the turnover of the cell as far as telomere shortening goes. And if a cell is turning over and it lacks any kind of mechanism, telomerase, to keep the end, the telomeric sequence, it will lose telomeric sequence with each round of replication. Now, there's been some studies in, like the brain, actually telomeres get longer, and there's not much, if any, turnover. So I guess the point I would make is that cell type that your clear tissue is very important. Okay, so because we can get blood easily, it's accessible. It's non-invasive. We can, you can do large studies on blood, and we've done those too. But if you're looking at breast cancer or something like that, looking at the blood might not reflect what's really going on in the breast, possibly. And so it just makes the studies much more difficult. So you do things like Alan Meeker's work at Hopkins, where he's actually looks at the tissue sections and does this Q-fish type method to measure telomere lengths, which is, and so you really zero in on that particular cells and those particular cells, and you can get something interesting out of that kind of work. It's just very hard to do that on a large scale. So I think your question is very interesting. It's just a little bit too broad, in the sense to get at, and we could talk more about it afterwards too in specific cases.
Did you ask, I think you asked a question about what are the telomeres doing in interphase? Did you ask that?
Yeah, what I said was, the concept would be potentially they're parked at the perinuclear membrane, or they're potentially localized in certain regions. So maybe in David's study where he's asking, is chromosome 1 attaching to chromosome 5 versus chromosome 6 or 10? Is that a relation that has a contribution by chromatin architecture and nuclear architecture? And virus that would be perinuclear would also potentially be able to be interacting in those same zones.
So Sabina Mai has been studying this. Are you familiar with her work up in Canada? The last time I read her work, what she had been describing is that during interphase, like you were talking about, the telomeres get together into like a rosette, and they sit there, and they're very protected. Am I correct, David?
There's actually a lot of disagreement about that. So,
but that's what she reports.
There are
right?
some species, that have been known to get into these clusters or bouquets or whatever you want to call it. But there's a lot of controversy right now about whether or not that's happening in humans or not.
And whether or not it's in a rosette also has to do with how it interacts with its environment and whether it's attached or whether it's moving, etc. So I think the point that you make about chromatin structure, movement, has a lot to do with what's happening with the telomeres and what chromatin domain they find themselves in.
No, it's a very, very interesting area. I think we just like the tip of the iceberg analogy. We sort of know most things. We just know a little tiny bit, and that's certainly a direction in the future. But a very good question.
Quick couple of questions. I'm Margaret Pratt from the EPA. Referring back to Dr. Sandler's discussion of the twin studies where there was no difference in telomere length seen between mono and dizygotic, I was wondering if that included dizygotic of different genders. And then also, secondly, has there been an examination of twins that were born from natural conception versus twins who were born from in vitro fertilization?
These are twins who were born a long time ago, before there was IVF, and they're veterans, and so they're all, and this is World War II, and so they're men, so they're all same sex.
Right, but no follow-up studies later with the newer technologies.
You may know, but I don't know.
That was a study that we did a few years ago. So there were World War II veterans. Most of those men have passed away. I mean, they were really at the terminal stage in the late 70s. Some older, most of them passed away. So, but it was brothers. But it would be very interesting, to look at brothers and sisters. And
I have a question from the mail in, and basically the question is whether there seems to be the telomere lengths are more heterogeneous in laboratory mice than in humans. Does anybody know why this is the case? Generally, we see more heterogeneity in humans in other biomarkers.
Well, lab mice are kind of strange. They're inbred, and they have these very long telomeres, and there is a lot of heterogeneity in the telomeres, but because it's run out in a certain jail, it just sort of all clumps up at that point. But if you do a different kind of analysis where you separate, there's a lot of heterogeneity within the mouse. Wild-type musculus has human-sized telomeres, so it seems to be as a result of inbreeding. It's also really hard to the telomere biology in mouse is really different than human. Telomeres is pretty much expressed all the time in every cell. You have these inbred mice lines which have very long telomeres. So there's some very interesting things you can do in mouse, but it's also difficult to extrapolate the findings that you find in mouse with human, especially in cancer studies.
Downstream consequences are also different because mice are wired differently than humans in terms of P53 and RB. Those pathways don't connect in the same way. And so since that's a major readout of telomere dysfunction.
Thank you so much. I would just take this opportunity to close the session and to thank every speaker in the morning who have given an all very stunning talk and very insightful discussions here from the audience, and the questions are just outstanding. So I have to say that it is a very successful conclusions for this session. Thank you all.